My name is Cyril Berthet. I'm the head of the pharmacoimaging unit here at Oncodesign, and I'm also the head of the PharmImage group, which is a local consortium that includes chemistry, pharmacology, and clinical imaging. So today, I will introduce you some questions, some discussion about the drug biodistribution and efficacy, and how to manage radiolabeling of molecules and how we can make the difference in your program. So I'm going to start this presentation now. The main objective of this webinar is, first of all, to show you how to design and implement radionuclide labeling of your chemical and biological molecule of interest. Then we will go through some of the biodistribution of the therapeutic drug, how we can use imaging to follow up your drugs. Then we'll move to another topics, that's how to develop a translational nuclear medicine biomarker, just for the drug development, but also to really be a companion of your drugs much further than the first step of the preclinical development. And at the end, I will show you just a few slides on the evaluation of targeted radiotherapy with a specific radioisotope. So the first point of my talk is about radiolabeling, how to select radioisotope, how to select chelating agents, and the radiochemistry itself. When you have to radiolabel molecules, you have to consider the size of your molecules and the chemistry feasibility. That will clearly define the choice of your radiolabeling methods. It depends also on the in vivo stability of the molecules. So it's a combination between the size, the stability, the in vivo PK, that you will decide which radioelement you want. For small molecules, usually we will use a covalent bound radionuclide, because if you use larger if you do bioconjugation, it will clearly affect your small molecules. So this is not the proper way to follow up. So for small molecules, we will work usually with C14, tritium, iodide elements. And fluor is also one of the best one to use. It's really depending on your PK and the half-life of your compounds. I will show you a table right after when you can compare the half-life of your compounds and the half-life of the radioisotope. And for larger biomolecules, we usually use the bioconjugation with the chelating agents. This is what I'm showing here. So you have a bioprotein or an antibody that you will add a linker, which is a kind of cage, and chelating agents where you can add your metallic radionuclide. In this case, we usually work with copper, gallium, indium, lutetium, technetium, this is or zirconium. This is the main one we can use. On the next slide, there is a table of the major radioisotopes that are of interest in nuclear medicine. Depending on the application you want to be, it could be PET imaging, SPECT imaging, or simply biodistribution without imaging. Will we pick one or the other radioisotope? Depending on the PK of your compound, as I just mentioned, if you have a PK of a compound that is 5 days, you don't want to use fluor that is only 2 hours of half. When depending on the application, we will pick the proper isotope. So we can work on PET imaging, SPECT imaging, or simply biodistribution without imaging. Depending on the half-life of your compound, we'll pick one of the best isotopes. So for fast compounds, fast PK, we'll use gallium or fluorine, where you have a short PK, short half-life, 1 hour or 2 hours. Or if you have an antibody that has much longer half-life, usually much longer PK, zirconium or copper that stay over 3 days or 12 hours are the best to be picked. For SPECT imaging, one of the standard one is technetium, that is 6 hours. Indium is a very good one for most of the protein or antibody because you can go up to 2.8 days with a signal. Iodine is one of them. And lutetium is also a good one, but mainly when you want to go to therapy. If you don't need specifically imaging, then we can use QWBA, autoradiography, and carbon-14 or tritium, iodine-125 are usually very good, and you can go up to many, many years with these compounds to follow up your compounds. So this is always a matter between the half-life of your compound, the time you want to image, and the half-life of the radioisotope. Then there are another kind of isotope that are it's with different what we call beta emitter, and they are of interest for radiotherapy. One of the main one that is used currently is iodine-131 or lutetium-175. Half-life of this radioisotope are between 6.7 days or 8 days respectively. One is covalently binding and one is through chelation. Just to illustrate the point of chelation, chelation is we will add a bifunctional chelating agents, meaning a cage where the radioisotope will be bound, will bind into this cage and will be fixed to avoid any release in the blood or in the body of animals. And other parts will be fixed either to the NH2 or SH group, to the lysine or to the cysteine, and depending on your biomolecules, and the radioisotope will pick one of them. The classic chelating agents are DTPA, DOTA, or DOTAGA, but many others are available. GFO will be used for zirconium, for examples. And again, this is depending on the radioisotope and also on your biomolecules if you want to be fixed on an NH2 or SH group. Depending on your question, depending on your compounds, our expert will help you to define which chelating agents you want and which radioisotope you will need. When you work with radioisotope, you need specific environments. And here we have access to 3 types of laboratories for radiolabeling. One is, of course, for bioconjugation, and when you work with metallic radionuclide, most of them you can be, this radioisotope, the metallic radioisotope can be delivered to you as they have long half-life without major issue, except for gallium, that has only 1 hour of half-life, and you need to have a generator in your lab, which is the case here at Oncodesign. And there is another major radio element that we'll use for covalent blinding is the fluorine 18. And in such case, you need a cyclotron, and just next to the cyclotron, you need a laboratory to process the fluorine. And here in the picture, you have, here is our research lab, and the other part is for a GMP lab to prepare clinical batch of radio tracers. So this kind of environment is very important and very controlled by all national organizations, each country that's managed the radioactivity. There are 2 kinds of radiolabeling. One is covalent and one is bioconjugation. So here I just show you very simple example for chelating agents for covalently binding. Fluorine is one of the example, but as I mentioned, carbon-14 is another one. Carbon-11 could be also one for very short half-life. Tritium, and you will do specific chemistry, covalent chemistry. Here is an electrophile substitution of the fluor. For bioconjugation is an example of trastuzumab. We will bind with the chelating agent, and after that we will integrate, we will add it into the reaction, the indium to the compounds, the bioconjugated compounds radioactive. There are some controls that need to be performed during any labeling process. For bioconjugation, we have to measure the ratio. What is the proper ratio between the chelating agents and the biomolecule? Usually a ratio 2 to 1 is what we try to reach. especially with the random radiolabeling. If you have many cysteine or many lysine, it's good to control this ratio to avoid any impact on your biological activity. So usually we try to reach a ratio that is around 2. We have to check, of course, the linkage of the radioisotope, covalent or bioconjugation, and we have to check the biological activity. Commonly, it happens that depending where your chelating agents, where your molecules your radioisotope will be bound, that it may affect your biological activity. So it's very important to control that before any in vivo experiment, any in vitro or in vivo experiments. Other control that is possible, you can check the isotonicity, the pH, the sterility. This is mainly when you want to inject to animals. And also, for example, protein aggregation. Sometimes when you add chelating agents with on some antibody, you can drive some aggregation of your antibody. So it's important to check before any in vivo experiments. So before I'm moving to the next steps, the biodistribution nuclear imaging, just want to inform you that a new poll has been posted by Krista. So if you have regarding if you have any forthcoming project related to our current webinar, feel free to answer this poll. So coming back to the presentation, so now I'm going to switch to the biodistribution itself, where we can evaluate the evaluation of target expression and molecule in vivo biodistribution. Here is a standard design of a biodistribution study. It involves chemistry, biology, preclinical imaging, and data analysis. So you need several expertise to be able to run properly such experiments. So first of all, radiochemistry. You need very specific experts to design the chemistry reaction, to prepare them, and to evaluate the quality of your bioconjugation or covalently binding. And then you need to have access to in vivo model or in vitro model if you want to do any in vitro experiments. So you need to have access to a pharmacology group. Then you need imaging experts, physicists to manage the equipment, and then to do the analysis of your image. What we can, when we perform such a study, we will do first batch, it's what we call a test batch on your, just to validate the procedure on your compounds. We will check the biological activity of your compounds, and then we'll produce complete a production, what we call a production batch for the following experiments. There are different controls that we can, different assay, immunoassay, ELISA or any other assay, phenotypic assay that we can use to validate the biological activity. Radiolabeling will be checked by RTLC, and for the in vivo model, at the time of the biodistribution itself, we will repeat the radiolabeling, validate by RTLC T-ready labeling, and perform the imaging. So a biodistribution study itself is usually between 24 and 72 hours, sometimes 5 days. But the whole study, if you start from chemistry up to the end of the imaging analysis, is between 10 weeks and 24 weeks. It's really depending on the models, really depending on the difficulties of the study. So keep in mind that the biodistribution is a short study, but the whole process it will be 10 to 20 weeks. This is the kind of data that you will get. Here is an example of a SPECT molecular imaging of a BT4474, which is a breast cancer model that has been subcutaneously engrafted. This tumor cell line overexpress HAR2, which is the target of trastuzumab. So we'll perform a time point at 24 hours after injection, at 48 hours, and at 72 hours. So in such case, you need depending on the PK of the antibodies, you need a radio element that allow you to do imaging after 72 hours. Indium is one of them. And you can see that the background is decreasing over time. You have some unspecific binding at first, and then it's getting clear, and you have a very nice image at 72 hours. We always correlate the imaging with ex vivo experiments. Will be done at each time point or only at the ends, and we correlate the gamma counting on each of the organ with the SPECT counting quantification that we can perform live. It is very important to correlate both of these measures to be fully accurate. Then we can use PET, PET scan with other radioisotopes such as zirconium, for example. And here is one example. Actually, this machine here is not really only a PET machine. It's also a new prototype. It's a PET MRI, which is very interesting because you can add the morphological imaging up to the functional imaging. So, and PET MRI is often much more precise in terms of morphology than PET-CT. This new prototype has been developed in collaboration with MR Solution, and that is now available for sale, but also we've been developing expertise of using this machine. And here there are two kinds of experiments. One is a tumor PET MRI, when you can really see the difference, the morphological in organs and the tumor, and for cardiac also imaging is very important. You can combine FDG and MRI, cardiology by MRI sequence are very interesting image you can get. Then there is the hot topic, which is the tracking or the trapping of immune cells. As you know, in the tumor site is not only tumor cells, but there is a lot of immune cells that infiltrate the tumor. There are different approaches that usually people working with immunocheckpoint are interested. The first one is to just follow up the immune cells. Where are the immune cells? So actually, we are developing a program that's in Biomarker to radiolabel CD8 to track tumor T-cell infiltration. There are other targets that we are working on. This is a work that is ongoing and should be complete by the end of the year. And we will work with our humanized mice or syngeneic mice, depending if the CD8 is targeting the mouse or the human T cells. This could be a very interesting approach to target T cells. Another approach that is commonly asked to us is to biodistribution of bispecific molecules. It's very common to develop now bispecific molecule, one target being on the tumor cell and the other target being on immune cells to trap them into the tumor. And in such case, we will label the bispecific molecules and we will compare them, the biodistribution in normal mice bearing a tumor or not, versus humanized mice with T cells or any subpopulation, PBMC or any subpopulation. And so we can compare within the tumor if indeed the biodistribution is different in presence or not of humanized immune cells. This is a very interesting approach to really determine if you trap immune cells within the tumor. Here is just an overview when you want to do a biodistribution. Usually the first step could be fluorescence if you want just to screen your different antibodies, different fragments, whatever your biomolecule is. This is the simpler way to screen molecules, although cytochrome might clearly impact the biodistribution of your compounds. Anyway, this is the cheapest and fastest way if you have many antibodies to screen. Then you can do a complete distribution with SPECT. Usually really easy to do. Technically, there is not many challenge and can work on most of the compound, most of the antibody. If you want to go to clinic and you need a biomarker, if you want to go further in your development, PET usually is better than SPECT in terms of quantification, in terms of localization and sensitivity. So using PET with zirconium, with copper, with gallium, is usually will improve your readouts. And if you have access to a PET MRI, of course, you will gain another layer of sensitivity and imaging readouts. Now I'm going to switch to the clinical biomarker. What to do if you want to develop a radio tracer up to the clinic, up to the market. This is the flowchart that we have developed here within our internal research program in Akilim. So first, you have to select your target and your compounds with very selective compounds. Otherwise, you can imagine that your background and your interpretation of the model the imaging cannot be done. You have to perform the radiochemistry. Then you do the binding assessment in biological sample to determine your Bmax. So after the biodistribution and the proof of concept that your molecule is reaching your targets, you can move to a clinical study, phase 0, phase 1, and later on, if you want to access market, to phase 2. So here we see an example of one of our programs. That's the goal was to develop an EGFR PET radiotracer. First of all, to address the activity of EGFR in tumors, especially in lung tumors, and under treatment, the signal was expected to decrease, and after resistance, when the EGFR is reactivated, usually through a mutation, I don't want to go into details, and there are reactivation of EGFR, and you can get back the signal. So the purpose was really to follow up the therapeutic response of an anti-EGFR with this kind of tracer. When you want to develop a PET radiotracer, there are specific characteristics that you have to keep in mind. Your target product profile will be different than for a therapy, and we select different criteria that we judge that are very important. Pharmacologically, we consider that the Bmax, the maximum binding over the KD should be over 10, and the selectivity from your main target should be around between 30 and 100 times with the off-target. The non-specific binding is very important, and you want at least 5% of free fraction of your compounds, and CLOC-P should be lower to 3. Then if Bmax is very low, if there is very low target, it will be difficult to have to get a signal. So when you start to have your molecule and you do the labeling with F18, you can compare with your cold compound F19 and do KD determination and specific binding. So you will have a slice with different concentration of your cold compound that you compete with your radioactive compound, and you can determine the specific binding and the KD. So in this case, with the KD that were above 50 nanomolar. Then you move to in vivo study. Here in this experiment, we have a rat with 2 tumors, one expressing EGFR that is not activated with no mutation, and one that is expressing EGFR with 1 mutation. And you can notice that in this case of a radiotracer, we have a very specific binding of activated EGFR. This was correlated with immunochemistry with phospho-EGFR. When you have phospho-EGFR, this kinase is activated, and this is what we have seen in vivo. And if you measure the tumor over muscle ratio, you can see there is a large increase of this ratio. So this is the different path that you have to go through when you want to develop a radiotracer. Then if you want to go to clinic, that's another story. You have to prepare the GMT synthesis. You have to do cold compound specification, formulation, radiochemistry, radiotracer lot production for your DMA file, preclinical toxicity, of course, the regulatory file, and the clinical study itself. This is not the purpose of this webinar to develop multi-stage. It's just an overview of what needs to be done before you can go to phase 0. And in our case, of our EGFR tracer, we went to phase 0, and this is the design of the phase 0 we did. We first administrate our compounds in 4 patients that carry the EGFR, and then in 6 patients that do not carry EGFR. And we made the comparison between the two groups to determine if there was specific binding or not in lung cancer. And this is one of the first results we get. When you compare the FDG standard image profile of a lung cancer, this is what kind of image you get. With our tracer, it was very specific focus, and so you can clearly determine there was a difference between FDG and probably this is tumor cells that do express the mutation, where here it's more rather the whole tumor plus inflammation. And in our design, there was a first step before the treatment and one was after the treatment. And you can see that there was no more signal after the treatment, meaning the patient was still under treatment here, and meaning that there was a saturation of the EGFR site, and probably the patient, at least the drug is going to the tumor, and most likely the patient will respond. This is just a case report, and the full clinical data of this study will be presented at the next EANM Düsseldorf. Before I'm going to the last part of this talk, there is another poll that has been put on the left part of your screen, about what make you choose us amongst other CROs. So I would let you answer these questions. So the last part of the talk is about targeted radiotherapy. We have to pick to do the radiolabeling with therapeutic isotope and to measure the treatment efficacy. So again, there is very specific expertise you need to do that. This is a very promising approach to cancer therapy, and it has been now validated with different clinical studies. And recently, for example, Lutathera, which is a dotatate that is bind to lutetium, peptide dotatate that is bind to lutetium-177, has been approved in Europe and in US for GEP-NET tumors, for the gastroenteropathic neuroendocrine. Dotatate is in fact a ligand for the somatin receptor. So when you want to develop such therapy, first of all, of course, you have to select the models that express the target that you have chosen. It could be targeted for an antibody or for a peptide or a small molecule, whatever, but you need carefully to select the model that expresses your target. Then you need to do the bioconjugation of the radiolabeling. Using such isotope requires, some expertise and some level of radioprotection, because the beta emitter and the half-life of this radioisotope is more complicated to manage than that fluor or the short-life isotope. You will perform a first escalating dose to measure the tolerance of the pure preclinical models to this peptide and to the specific activity that you will use. Then you will determine the efficacy at single dose. Then, using a different ratio of your molecules on radioisotope, you can also play on the efficacy between the cold and the hot part of your therapy. Then you can play with the fragmented dose using the same amount of radioactivity, but over multiple treatment, like it is done in clinic, and not one single treatment. And very commonly now, people that work in this field will develop in parallel radiolabel diagnostic tools with gallium, copper, zirconium, and this is used, in fact, in clinic to select the patients that will express the target in the tumor. I'm showing you just two examples that we've performed. One is with yttrium-90 and one is with lutetium-177. So you can see in both cases of this therapy, we observe a very strong activity of this kind of therapies. And I think this is one of the reasons that is getting more popular now that we can manage toxicity of such therapy, and it's much more targeted to the tumor. Of this webinar, I show you how the radiolabel molecule can support your preclinical and clinical developments, the biodistribution of your compound in rodents, non-human primates, and human. I didn't show here non-human primate because of time, but this is also an expertise that we can provide. The co-distribution with human cells, this is something that is getting more and more important. Development of biomarker to select patient population. And our expertise has been built through different consortium that Oncodesign is leading. PharmImage, first of all, which is our local network, as I mentioned at the beginning of the talk, that go from chemistry to clinical imaging. We have experts in radiochemistry, in vitro and in vivo model, preclinical imaging, clinical imaging, and development of new imaging agents. And we have multiple collaborative research programs within this consortium. One is imatinib. This is where we developed the Egfr radiotracer. Was one IMAPI when we developed the PET-MR prototype, and Biomarker, which is a new one where we'll work on the tracking of immune cells. And so we can offer to our client and to our partners several kind of pharmacoimaging services going from chemistry to up to radiotracer development. This is done for me, and I welcome any questions that you may have. So I'm going to the chat and looking at the question. So what are the chelating agents already accepted by the agencies? Yes, so there's a question from Pascaline. Yes, some of these chelating agents are already accepted by agencies. The TPA and DOTA are some of well-known. There are some clinical trials that have been done with GFO. And here we are working with a company named Chematech, which is part of our PharmImage consortium here in Dijon, and they are producing some of these chelating agents under GMP condition and can be used in clinic. And then there is a question from Christophe. Was the EGFR tracer used in patients a full antibody or smaller construct? So here our EGFR was a kinase inhibitor. So it's a small molecule that were covalently bind to fluor. So it's we are clearly measuring the activity of EGFR because of small molecules will bind to the ATP pocket of EGFR. So it is not an antibody or Fag fragments. Then there is another question from Pascaline. DOTA chelation require harsh condition in terms of temperature, pH. Will be Nodaga a better chelating agent? It's depending on your radioisotope you want to use. It depending on your biomolecules. When we start to work with the new molecules, we have kind of a questionnaire that will be sent to you that will let us know what kind of pH temperature we can use. And indeed, we will choose the chelating agent that will fit to the chemistry reaction we can use with your molecule. So yes, Nodaga could be better than DOTA depending on some of your condition. And we can help you to define which one is the best. If there are no more questions, we can close this webinar. Thank you to everyone for attending this webinar, and you can download all the slides and have a replay. And of course, you're welcome to contact me directly at this on our website or on PharmImage or through LinkedIn. You have my emails here. So if you have any questions regarding radiolabeling and preclinical pharmacoimaging, please feel free to contact me.